Good morning, Jackie, Barbara, everyone in the audience. So I'm going to kind of power through this because I have a lot to cover. These are my disclosures. I'm going to mention the PI3K inhibitors, and then I'm breaking up the BTK inhibitors into covalent and non-covalent. On the covalent, I'm going to talk about everything but tirabrutinib. That actually doesn't have any trials that are currently accruing on clinicaltrials.gov, so I don't know if that's being developed further. And the non-covalent, I'm only going to talk about the Arcule and the Vecabrutinib because those are the only ones that have clinical data that's available so far. So idelalisib, as you know, is a PI3K delta inhibitor. The relevance of the delta being, as you can see, it's very specific for hematopoietic cells and the most selective of all of them. The trial that led to the approval of idelalisib actually led to the approval of idelalisib in combination with rituximab. So it's not approved as a single agent, it's approved as a combination. And that was this randomized trial in very, very refractory patients, really some of the most refractory patients I've ever seen, where they received rituximab and idelalisib versus placebo and rituximab. The primary endpoint was progression-free survival, and later crossover was allowed. The progression-free survival was significantly longer with idela and rituximab, again leading to the approval. It was about 19.4 months. And very importantly, there was no difference in PFS based on mutation status, which we see so far with pretty much all the small molecules. But somewhat different than the other ones is there was no difference based on 17p or p53 status, where the progression-free survival was overlapping at 20 months. So it's a very active combination in relapsed CLL. One of the issues, of course, is what we call the itises that are thought to be perhaps on-target effects from inhibition of PI3K delta, which then inhibits Tregs, potentially allowing a cytotoxic T cell population to emerge. You actually see this in less heavily pretreated patients, probably because they have more intact T cells and are better able to mount a cytotoxic T cell response. And ongoing work is trying to identify some of the predictors for this toxicity, where there's a low incidence of pneumonitis, more transaminitis, which obviously is not a big problem, but some late colitis, which can be problematic. Duvalis was just recently approved in the US. I don't think it's approved in European Union yet. This is a little bit different. It's a PI3K delta gamma inhibitor. So this inhibits two isoforms, delta and gamma. And gamma may be particularly important in inhibiting T cells in general, which is why this is also being developed right now in T cell lymphomas. The trial that led to this approval was actually patterned on the RESONATE trial, which, if you recall, was a randomized trial in relapse of, here in this case, it's Duvalis giving at the dose of 25 milligrams BID compared to ofatumumab as a single agent. The primary endpoint was progression-free survival, and again, crossover was allowed. And what you can see is that the median number of prior regimens here was 2, so not as heavily pretreated as in the original RESONATE trial. And the trial, which was called the DUO trial, met its primary endpoint of progression-free survival compared to ofatumumab, and this again led to an approval. In relapsed CLL, there's also a separate trial that led to an approval in relapsed follicular lymphoma. So in the US, this is available for both. Also, there was a much better PFS with the 17p deleted than with antibody alone. In terms of the toxicities of interest, again, the itises, as you would say, that we see with idelalisib, these are the incidents here. In general, my perception is that it's the same spectrum of toxicity, but somewhat less frequent. Now, whether that's related to the fact that the number of patients who've been treated over time is a much smaller number than the number have now been treated with idelalisib, and there's much more aggregate data for idelalisib, or whether this is real, I think will remain to be seen. One explanation for why it could be real is, as I mentioned, the gamma component, the isoform, really is targeting all T cells. So you not only may be inhibiting Tregs, but not allowing so much of a cytotoxic T cell population to emerge. But again, we'll know more as we go forward with this agent. And then the third PI3K, which is not available yet, is umbralisib, which, as you see, has a very different structure than idelalisib and Duvalis. And this is also a PI3K delta inhibitor like idelalisib. The phase 1 study was just published last year, and this was done in a variety of malignancies, including non-Hodgkin's lymphoma, CLL, as well as T-cell lymphomas. And umbralisib was given at various doses. The target dose moving forward is 800 milligrams of the micronized formulation. And the primary endpoint here, obviously, in a phase 1 was safety and efficacy. So this was the trial population that you can see. And so you see that it really was a variety of different diseases. And I'll focus in on the CLL because that's, of course, the one of interest to us here. The median number of prior regimens was 3. In terms of the toxicity, this does appear to have the least amount of toxicity. So you notice that the itises are reduced. This population is all relapsed, so no frontline data in terms of whether that toxicity may be higher in the frontline setting like we see with idelalisib, but really very low incidence of some of the toxicities that can be much more problematic with idelalisib. And if we focus in on the CLL population and the assessable for activity was 20, we see that the overall response rate was 85%. Duration of remission actually had not quite been reached. And the median PFS is actually about 7 years in this group. So another PI3K with very potent activity, which may have a more favorable safety profile. And has been, the registration studies are accrued, and so potentially in the US, we could have approval as of next year. Now, if we switch over to the BTK inhibitors, obviously ibrutinib is the prototype here, and we now have the longest follow-up with this agent, which is 7 years. And this was data that was at ASH by John Bird. And so let's look at this data because this is really good follow-up and the longest follow-up we have with any of the small molecules. So I just want to make a small point about the patient population. Okay, the median number of prior regimens was 4. These, I think all the investigators would agree that these were some of, at the time, the most refractory patients that you could imagine. There were actually patients where the option was really to go on this trial or in some cases to go to hospice because there was nothing left for these people. So obviously the median number of prior regimens of 4 communicates to you that they're pretty refractory. But what I think really gives you an idea for how bad the population is not the median hemoglobin and platelets, but focus in on the range. So if you look at the range, so let's look at the lowest hemoglobin here was 6. Obviously, you're going to be transfusing somebody. So the point is they were transfusion dependent. Look at the lowest platelet count was 2. So the point I'm making is this is one of the first trials that actually allowed patients who are red cell or platelet transfusion dependent, because up until now, most trials, of course, were chemo-related, and those patients would have had more myelosuppression and not been allowed on the trial. So I think that kind of speaks to how refractory some of these patients were. The response rate was great across the board. I'm not talking about that small frontline cohort over the age of 65. Let's just focus on the relapsed refractory. Very high response rate in this 101 heavily pretreated patients. It was actually very exciting to see this in the early days of the trial because, as I said, these were very, very refractory people. But to be honest with you, I think most of the investigators just kept waiting for them to relapse, thinking, well, this is great, but how durable is this really going to be in this really very refractory population? And then, surprising to say, they weren't relapsing, which was very heartening, but I think, quite frankly, very surprising. And we now know that the median PFS in this highly refractory group was 51 months. Really was stunning at the time. I think it's fair to say you would have been lucky to get 6 months out of anything if you could have even come up with something to give a lot of these people. We do see that the 17P continues to be a high-risk group where the median PFS was 26 months. And the 11Q also did a little bit worse in this trial with a median of 51 months. I'd just like to point out that the median PFS for both of those refractory groups was better than any published frontline data for either 17P or 11Q at the time. There is emerging data that actually in the less heavily pretreated, particularly the frontline patients, the 11Q actually may be morphing into a favorable group. And in some cases, they're doing better than the non-11Q. This, again, was a very refractory population. Still no median overall survival for this population, which is very striking. What about acalabrutinib? We have acalabrutinib in the U.S. approved for mantle cell lymphoma, not yet approved for CLL. Both acala and zanubrutinib have the potential benefit, as you can see from the left-hand part of this slide, of having less activity against some of the other kinases that are targeted by ibrutinib by having a higher IC50. And the importance of this, of course, is that that would make it less likely to affect these other kinases, the relevance being that the feeling is that these other kinases may be responsible for a lot of the toxicity that we see with ibrutinib. So, for example, in boys born with X-linked agammaglobulinemia, which is due to BTK mutations, there are no bleeding issues in those patients. So again, some of the toxicities like AFib and bleeding are actually felt to possibly be related to inhibition of some of the other kinases. So what we see in the relapsed refractory trial was it was published by John Bird originally in the New England Journal and then updated at ASH in 2017. The target dose, the original dose was daily dosing, but then some of the BTK occupancy rates suggested higher levels of BTK occupancy with BID dosing. So later on, all of the patients were switched to 100 milligrams BID, and that's the dose that moved forward into all the pivotal trials. So on this relapsed refractory cohort, there were 134 patients. Median number of prior regimens was 2, so not quite as bad as in the phase 2 data for ibrutinib that I showed you. 17p deleted were about 23%. So although these were not quite as refractory as the patients I showed you on the phase 2 ibrutinib, some of them were really refractory. So this was actually the very first patient that went on the trial. And this was a patient that I had been holding on to for a couple of months, waiting for this trial to open. And the reason I was holding on to this patient is she was red cell and transfusion dependent. The ibrutinib trials were closed. I had no other trials for her. So the day after she went on acalabrutinib, her platelet count came back 0. Now, I actually also treat acute leukemia. It's very, very rare to see a platelet count of 0. Usually they search the slide and they can find, 1 or 2 platelets. This is the only CLL patient I think I've ever seen whose platelet count came back 0. And I am very happy to say that this patient had a great response and became transfusion independent. So this is the median PFS. You see, what you're seeing here is the trend I just mentioned to you. The better curve, or the highest curve, if you will, there, actually the 11q deleted patients. The blue are all comers, so nowhere close to a median. And the red and the green are still that higher risk category of the 17p deleted, p53 mutated, or complex karyotype. So those groups still tend to be higher risk, even with the newer small molecules, which are so much more effective in those patients than chemotherapy. In terms of the toxicities with this phase 2 experience, there was 1 episode of grade 3 bleeding, and other AEs of interest that we see with ibrutinib included hypertension at 13% and AFib at 3%. So, of course, some of that is related to the follow-up, which in this case was about 2 years, because we know with ibrutinib that hypertension continues to develop over time, suggesting, of course, that that number could go up. AFib can occur late, but I'd say it primarily occurs early. So there's certainly a suggestion here, I would say, that perhaps some of the significant toxicities are reduced. There is a head-to-head trial of acalabrutinib and ibrutinib in relapsed patients with 17p deletion or 11q deletion. We have not yet seen any data from that trial. The trial that will likely lead to the first trial that will lead to a registration for this drug is a trial that was presented at EHA by Paulo, which was a randomized trial of acalabrutinib versus rituximab Bidela or BR physician choice. I'm not going to talk about this trial because you're actually going to hear this trial presented later this morning. The other BTK inhibitor that's a covalent binder that I mentioned is BGB3111, which is now called zanubrutinib. Again, it has a selectivity for various kinases that are targeted by ibrutinib. The CONTAM presented the phase 1 data in 2016 at ASH. That has actually not been updated, but will be updated at ASH this year. What I'm showing you in the blood reference there is the phase 1 paper. So that's the first clinical paper published, but it has a variety of diagnoses in it other than just CLL. So here I'm just focusing in on the original CLL data. So 46 relapsed refractory patients. Median number of prior regimens here was 2. And I think this makes sense because as you go into development of new molecules, and there's so many now, nobody's going to give 4 or 5 lines of chemo to patients anymore. So we are seeing a very changing population in some of the trials now compared to what we saw in the original ibrutinib trials. So one of the things that this trial did, which is a little bit different, is it looked at BTK occupancy not only in the peripheral blood cells, but in the lymph nodes. And this might be important because there is some data that patients with bulky adenopathy are not as likely to have deep responses to ibrutinib. And so here they were actually able to, in about 30 or 40 patients, get lymph node samples and measure BTK occupancy within the lymph node. And you see that it was quite good at the BID dosing. And so again, this one is moving forward at 160 BID. If you look at the bottom of the slide here, we're focusing on the AEs of special interest. And again, smaller numbers, shorter follow-up, suggestion that some of the side effects that we worry about, like AFib, are maybe reduced with this agent. Here's the efficacy summary. Very high response rates with this drug, 96%. Very few CRs. Nice improvements in cytopenia and progression-free survival. The median follow-up here was about 13 months, so relatively early, but very encouraging. The pooled safety data is over 600 patients, so this is just looking at adverse events. And again, now with a denominator of over 600, a suggestion that we are seeing some less toxicity for, again, some of the important AEs of interest. And finally, just a few words on the non-covalent inhibitors. The obvious potential for these is in patients with BTK mutations where you no longer have covalent binding of the BTK inhibitor to the CYS481, whereas if these drugs that bind in a different binding site would putatively be active against these refractory BTK patients who have a mutation. It does have some less toxicity. Vecabrutinib, this is the senesis agent. For some other kinases, you can see that it's quite potent on the right. You're seeing ibrutinib in a mutated cell line here. You're seeing ibrutinib, which is the black, which is the red, acalabrutinib, which is the red, black, which is ibrutinib, and then the blue curve, the most sensitive, is vecabrutinib. And all of these drugs so far are not only active against the resistant clone, but they're active against the wild type also. So the phase 1 study is underway. It's still quite early. This is the only dose level I'm showing you here is from a poster at EHA, which went up to 200 milligrams BID. If you focus on the right, there's 19 CLL patients. This is a very refractory group because, in fact, if you have CLL or a mantle cell to go on this study, you have to be refractory, I should say, to BTK inhibitors. So most of the people have failed chemo, and now they've failed a BTK inhibitor. So you can see on the right, 74% of them have p53 deletions. So again, a very refractory group of patients. BTK mutation in 44%. It's also being investigated in lymphoma, but lymphoma where the drug is not approved, they don't have to have failed a BTK inhibitor. Looking at some of the correlates to suggest there's activity, we see reduction in chemokines in the patients very early on. And there's now stable disease in 3 of 5 BTK mutated patients. Again, very early, no toxicity seen, still in the dose escalation phase. And finally, vecabrutinib, which is also very potent against both wild type and mutated BTK, is a non-covalent binder. You can see here that here we have a little more clinical data. This is the swimmer's plot. The one that you see that's the longest response over 2 years is actually a follicular patient who didn't have a mutation, but there are a number of patients there. And as you can see from the bottom of the slide, 4 of 6 patients with a mutation have achieved a response at 65 milligrams, and that's the dose that's moving forward. So this one is a little further ahead and has reached a dose. So to conclude, there's 2 FDA-approved PI3K inhibitors. One may be coming soon. Ibrutinib produces very sustained responses. I didn't show you this slide, but there's no new safety signals. We're not seeing late unexpected toxicity. Calabrutinib and zanubrutinib, if you're at least as efficacious, maybe more, remains to be seen, with possibly less toxicity. And the non-covalent inhibitors are promising agents to lengthen the life of BTK inhibitors. Thank you.